The Shroud of Turin has been swabbed, vacuumed, and sequenced, and every few years, a headline announces that scientists found DNA on it. The implication is always the same. Somewhere in those fibers sits the genetic signature of the man in the image, maybe even proof of who he was. But when you read the actual studies, the DNA tells a different story than the headlines promised. A stranger one, and a more honest one. Because the labs did find DNA, and plenty of it. It came from dozens of human lineages spread across 3 continents, from plants that grow on opposite sides of the world, and from animals and crops that could not have existed anywhere near first-century Jerusalem. Over the next 20 minutes, we will walk through exactly what the sequencing found, study by study and year by year. We will separate what the science actually established from what got bolted on afterward. By the end, you will understand something the headlines skip over. No laboratory on Earth can lift one man's genome off this cloth, no matter how good the technology gets. And once you see why, the real result turns out to be far more interesting than the myth it replaced. First, the object itself. The Shroud of Turin is a linen cloth a little over 4 meters long, kept in the Cathedral of Turin in northern Italy. It carries the front and back imprint of a man who appears to have been beaten and crucified. For centuries, many Christians have venerated it as the burial cloth of Jesus of Nazareth, and for many people it remains one of the most important relics in the world. Its documented history in Europe begins in the 1350s, in the French village of Lyre. Where it was before that, and how it got there, is argued over by historians, and no one has settled it. So why would anyone test a medieval-looking cloth for DNA at all? The logic is straightforward. Linen is woven from flax, but a cloth this old has spent centuries pressed against human hands, resting in human rooms, collecting whatever floated onto it. If any of that genetic material survived, it might say something about the people who came into contact with it, and about the places the cloth had been. The hope was genetics as a kind of passport for an object that traveled, its stamps waiting to be read. Here is the distinction that trips up almost every headline, so we are going to nail it down before we go any further. The DNA testing and the carbon dating are 2 different experiments, run for 2 different reasons. In 1988, 3 laboratories radiocarbon dated a thread from the shroud and returned a date range of roughly 1260 to 1390, which points to a medieval origin. That result has been debated ever since. The raw data was released and reanalyzed decades later, and the argument over it is still going. But hold that completely separate in your mind. Radiocarbon dating measures the age of the flax the cloth is woven from. DNA sequencing reads the biological traffic left on its surface, which can be centuries younger than the cloth, or centuries older, and tells you nothing about when the linen itself was made. They answer different questions, they use different methods, and one says nothing about the other. Conflating them is the single most common mistake in this entire story. Everything from here on is about the DNA and the DNA alone. The first major genetic study landed in 2015 in the journal Scientific Reports. A team led by Gianni Barcaccia at the University of Padua took dust that had been vacuumed from the shroud, including from the body image and from the edge that had been clipped for that 1988 carbon test. Then they sequenced whatever DNA was trapped in that dust. What came back was not what anyone was fishing for. The plant results came first, and they were already odd. The dust held DNA from a long list of plant species. Some were plainly Mediterranean, the kind you would expect on any old cloth from that region. But others traced to the Middle East, to East Asia, and even to the Americas. One cloth carrying plant genetics from most of the inhabited world. Already, before a single human sequence was read, the impression was of something that had been almost everywhere. Then came the human DNA, and this is where people started reaching for the word miracle. The team recovered mitochondrial DNA, a type of genetic material that survives far better than the rest because each cell carries hundreds of copies of it. It is also passed down only along the maternal line, mother to child, largely unchanged for generations. And they found it from at least 14 distinct human lineages. Not one person. 14 threads of ancestry, at minimum, tangled together in the same fibers. And they came from everywhere. Some lineages are common in Western Europe, others in Northeast Africa, others across the Middle East, the Arabian Peninsula, and the Caucasus. A cluster of the rarer ones had their closest matches all the way over in the Indian subcontinent. When the team ran the numbers, more than half of the human DNA matched Near Eastern lineages, and nearly 40% matched Indian ones, which left Europeans as a small minority of the sample. Hold on to that Indian signal because it comes back later, and there is a fascinating explanation for it. Now, a person hoping for a clean answer might read those numbers and get excited. Near Eastern DNA dominant on a cloth tied to Jerusalem. Surely that means something. But watch what the lead author actually said about it. Barkachia and his colleagues were blunt in the paper. The diversity was so wide, so scattered across the globe, that they could draw no conclusion about where the cloth came from or who had been wrapped in it. Near Eastern DNA on a relic that spent centuries in the hands of Near Eastern and Mediterranean people is exactly what you would expect either way, whether the cloth is ancient or medieval. The genetics were consistent with an object handled by people from many regions over a long stretch of time. The finding was almost anticlimactic. 14+ lineages, no origin, and no individual. So that is the shape of the 2015 result, and it raises a sharper question than the one we started with. If the DNA on this cloth comes from a dozen regions and most of the world's flora, then what the cloth has really been recording is contact. Every hand, every room, every journey, layered on top of the last. Which means the real question was never whose DNA is this. The better question is how many centuries of handling are stacked in these fibers. And a later round of sampling answered that, with one detail so specific, it leaves almost no room to argue. That detail is where we go next. Buried in that 2015 plant list was a clue that never made the headlines. Some of the plant DNA traced to species whose home is the Americas. At the time, it read like a footnote, easy to skim past. A decade later, a second team went back with far more sensitive sequencing and pinned down exactly which crops were sitting in the fibers. That list is the closest thing this whole saga has to a smoking gun. The crops they identified include corn, peanuts, tomatoes, peppers, and potatoes. Every one of those plants is native to the Americas, and each one carries a built-in timestamp. None of them was known to Europe, Africa, or Asia until after 1492, when the voyages of Columbus opened exchange between the two hemispheres for the first time. Before that, the Atlantic was a wall. Corn was domesticated in Mexico. Peanuts and potatoes came out of South America. A first-century farmer in the Eastern Mediterranean had never seen any of them, because the continents they grew on would not be reached by the Old World for another 1,400 years. So the timeline gives it away. In the 1st century, in a tomb outside Jerusalem, corn does not exist on that side of the planet. It cannot. Yet corn DNA is on this cloth. There is only one answer that fits. The cloth kept collecting biological material, century after century, long after it was made, through every hand and every place it passed. The contamination did not stop at some ancient date. It never stopped at all. A 1st-century burial cloth would not carry corn DNA. a well-traveled object that spent hundreds of years being handled, kissed, displayed, folded, and stored, absolutely would. The corn is a record of the 1,500 years of touching that came after the cloth was made. This is the frame that makes sense of everything else. The shroud has been behaving like an archive, or a sponge. It soaked up genetic traces from whoever and whatever it touched, and it held on to them, building the pile deeper with every generation. The New World crops are the receipt for all of it. They prove that the surface of this cloth is a running log of contact, built up across centuries, and that any single moment in its history now lies buried under all the others. That second effort was published in 2026, again in Scientific Reports, by many of the same researchers. This time they used a technique called metagenomics, which sequences every scrap of genetic material in a sample at once, without hunting for any single target in advance. You do not decide what to look for. You read everything first, and the sorting comes afterward. And they applied it to a set of samples that had been officially collected from the shroud back in 1978, filaments and vacuum dust that had been carefully preserved for nearly 50 years. One science magazine described the result as a cornucopia of contamination, and that is a fair summary. The samples held a dense mixture of life from every kingdom you can name. Human DNA, yes, but also DNA from cats and dogs, from cattle and pigs and chickens, from goats and sheep, and from the same crops we just walked through. There was even a strong signal from Mediterranean red coral, a species prized for jewelry and trade around that sea for thousands of years. One cloth, carrying a barnyard, a garden, and a seabed all at once. And it went smaller than that. The team reconstructed the microbes living in the fibers and found the exact bacteria that colonize human skin, the ones you carry on your own hands right now. That is not background noise. It marks direct human touch, over and over, worked into the cloth. They also found salt-loving microbes and molds, the kind that grow on an object stored for centuries in still, enclosed spaces. Even the germs were telling the story of a relic that was handled constantly and kept in cupboards and chapels across a long, sheltered life. Then there was the detail that says the quiet part out loud. When the team read the human mitochondrial DNA, the dominant lineage in several samples matched one specific living person, not an ancient one. It matched the scientist who had collected the 1978 samples in the first place. His own genetic signature was the loudest human voice in the entire mix, for the plain reason that he had handled the material directly. A kinship analysis confirmed it was him. If you ever wanted one clean demonstration of what this kind of testing captures, that is it. The most prominent DNA on an object is usually just the DNA of the last people to touch it. The researchers were careful, and their conclusion was measured. To guard against fooling themselves, they ran controls, including a modern piece of linen deliberately contaminated by a lab member, so they could tell handler DNA apart from anything genuinely old. The pattern of animals, crops, and human lineages, they wrote, suggested that much of the contamination built up in recent centuries, after the eras of Marco Polo and Christopher Columbus, when trade and travel spread these species across the world. They also flagged an unusually high number of tiny variations in the human DNA, the kind you only get when many different people leave traces on the same object. In their own words, that mixture challenges any hope of identifying the original DNA of the shroud. So the signal in these fibers is handlers and environment. Pilgrims, priests, conservators, scientists, and the rooms and roots the cloth moved through over hundreds of years. It is a genuine biological record, and a detailed one. And a record of contact answers a completely different question than a burial ever could. Which brings us to the idea that launched 1,000 headlines. Somewhere out there, the story goes, is Jesus's DNA, waiting on this cloth for science to finally read it. Some versions go further and talk about a divine genome, or blood that shows no human father, or an unusual set of chromosomes that ordinary people do not have. It is worth explaining, carefully and without heat, why the method itself makes every one of those claims impossible. This is about the limits of the tool, and nothing more. Start with what the labs recovered. Not a clean genome. Short, broken fragments of DNA, chemically degraded by centuries, and mixed together from dozens of different people and organisms all at once. Reading DNA like this is less like scanning a barcode and more like trying to reconstruct one specific book after every library in a country was shredded and the confetti swept into a single pile. You can prove the pile contains books. You can even name some of the authors. But you cannot pull out one intended novel in order page by page when its pages are torn into fragments and buried among a million others. There is a second problem, and it is even more basic. Almost all of the human DNA in these studies is mitochondrial DNA, a tiny loop of genetic code that sits outside the cell's nucleus, which every one of us inherits only from our mother. It carries no chromosomes at all. So any claim about an unusual chromosome count, or a missing Y chromosome, or an absent father, is describing information this testing never touched. One fact quietly dismantles the whole claim. Mitochondrial DNA is maternal for every human who has ever lived. Finding maternal line DNA on an object proves nothing whatsoever about anyone's father. It is precisely what you would expect from any surface that many people have touched, and it says nothing unusual about the person in the image. And there is the contamination itself. We just saw that the loudest human signal on the 1978 samples belonged to a modern scientist. So imagine a lab announced it had isolated one specific ancient individual from a cloth handled by thousands of people across 7 centuries. The first question any geneticist in the room would ask is a simple one. How did you rule out everyone else who ever touched it? With this material, the answer is that you cannot, and no future breakthrough changes that, because the information was scrambled together long before any lab arrived. None of that is a statement about anyone's faith. Whether the shroud is what tradition says it is, remains a question people answer through belief, history, and conscience. And the science here does not push on that in either direction. It offers no proof that the cloth is authentic, and no proof that it is forged. What the science does say is narrow and firm. This cloth cannot yield one man's genome. It cannot identify a specific person from history, and it never could. That limit lives in the method itself, and it holds no matter what anyone believes. So strip away the myth, and something real is left standing. The DNA does tell a story. It is a story about the cloth's travels, written without intention by everyone who ever handled it. The human lineages lean toward the Near East, which fits an object with deep ties to the Eastern Mediterranean, which brings back that Indian signal from earlier. The researchers offer a genuinely elegant idea for it. Fine linen, in the ancient world, was traded out of the Indus region, and the word for a shroud itself traces back through Greek to a fabric associated with that part of the world. So the Indian DNA may not be about who was wrapped in the cloth at all. It may be about where the raw linen was spun, thousands of miles from where the shroud became famous. The plants sketch a Mediterranean life, layered with crops that arrived from Asia and the Americas in later centuries. The animals, the cats and dogs and livestock, and that Mediterranean coral outline the rooms and regions the cloth lived in. Put it all together, and the genetics describe a well-traveled object, handled across Europe, the Mediterranean, the Near East, and beyond, over many centuries. That is a real finding, earned with careful science, a textile that has functioned, biologically, like a guest book. Thousands of people signed it without meaning to, in skin cells and stray hairs and the dust of the places it rested. Reading that guest book will never tell you the name of the man in the image. But it does tell you, in hard genetic data, that this cloth moved through the world and was touched by a staggering cross-section of humanity. The headline promised a person. The data delivered a journey. So back to the question we started with. Scientists find DNA on the Shroud of Turin. What did they actually find? They found that this is not the fingerprint of one man. It is the fingerprint of everyone who ever touched him, or touched the cloth in his name, across 7 centuries of documented history and however many years came before it. That is the honest answer, and it is stranger and richer than the myth it replaces. The myth offered a single, magical readout. Jesus' DNA, divine and unmistakable, waiting to be found. The documented science offers something no forger and no legend could have invented. A 4-meter linen archive of human contact, still legible after 700 years. On this channel, that is the pattern we keep running into. The documented record almost always beats the myth, and the myth was never the interesting part. Because one thread here is still hanging loose. If the DNA cannot date, the cloth and the carbon test that tried to has been fought over for decades, then the oldest real question is still wide open. Where was this thing before it appeared in France in the 1350s? And can any science recover it? That is its own story, and it is the one we are chasing next. Subscribe, and you will be here when we do.